Assalamu alaikum wa rahmatullahi wa barakatuh. Dear students, this is General Data Show Revision for Module 101. This module includes the cytology, connective tissue, epithelium, and the blood. Dear students, this is announcement that all the data show you should study it well because the revision include only the test, as well as you should study the label diagram. The most important chapter in histology is the cytology. Science deal with the cell. Why? Because the cell is the structural and the functional unit of your body. The cell is formed of nucleus and cytoplasm. There are elements in the body not formed of cell or not true cell as the RBCs and the platelets. The cell contains nucleus and cytoplasm. The cytoplasm contains organelles and inclusions. We said that the organelles are permanent, specialized, and vital. The organelles either membranous organelles or non-membranous organelles. This is an EM picture. This is an EM picture showing cell membrane of two adjacent cells. By EM, as you know, it is the stain for EM is osmic acid. When stain something appears black, when does not stain appear white, and in between grades of gray. These are two cell membranes. This is for one cell and this is for the other cell. It appears trilamellar because the heads are directed outwards of the phospholipid bilayer, which are stained with the osmic acid, and the tail does not stain. So it appears two dark layers in between the one pale, and between the cells there is the intercellular space which contains the fuzzy layer, which is the cell coat. So the structure outlined by the red rectangle is cell membrane of two adjacent cells. Visible character, it appears trilamellar. The blue star is the cytoplasm and the green arrow is the intercellular space. What is the importance of the cell membrane transport, which is either passive, does not need energy, active, needs energy, and the passive facilitative or simple, and the bulk transport endocytosis, either pinocytosis, receptor-mediated endocytosis or phagocytosis, and exocytosis. And the function of the cell coat. It does special stain for the cell membrane. Yes, it's stained with silver and pass by light microscope. This is an EM picture of two organelles. The star marked the mitochondria and the yellow arrow rough endoplasmic reticulum. In the data show slide, dear students, you should prepare a visible feature for identification. This is mitochondria. The visible feature of identification, the cristae. What is the cristae? It is infolding of the inner membrane of the mitochondria to increase the surface area. And this is the matrix, contains the enzymes, the catalyst, the RNA, and the DNA. The special stain for the mitochondria is iron hematoxylin and Janus green. The function of the mitochondria is energy production. The yellow arrow points to the rough endoplasmic reticulum, which appears as parallel flattened cisternae covered with ribosomes. On the surface, there is ribosomes, which are responsible for the cytoplasmic basophilia. If we said predict the cell in which these organelles present, it is a protein-producing cell. This is an EM picture showing mitochondria and rough endoplasmic reticulum. Here, visible feature, the cristae, the rough endoplasmic reticulum, presence of ribosome on the outer surface. This is EM picture of smooth endoplasmic reticulum. It appears as irregular shape vesicles, smooth, not surrounded with ribosomes. Here are electron-dense granules, which are the glycogen. As you know, that the smooth endoplasmic reticulum, one of its function, breakdown of glycogen. This is light microscopic picture stained with silver. Here, the organelle that's stained with silver is the Golgi apparatus, which either appears as granules or fibrils stained brown with silver, and in the nerve cell it appears perinuclear. and the green arrow is the nucleus. The red arrow demonstrates Golgi apparatus in nerve cell. From where I know that it's nerve cell, because the Golgi apparatus is perinuclear. Stained brown with silver, the green arrow points to the nucleus, which appears rounded and central. This is Golgi apparatus in secretory cell stained by silver. Okay, dear students, please look. I'm going to draw here. This is a cell. Okay, this is the cell boundary, for example. So this is the Golgi apparatus, which appears at the apex. Okay, and the green arrow points to the cell membrane, as we said that the special stain for the cell membrane is silver. And here is the apex. So the Golgi apparatus present near the apex in order to move a little bit the vesicles and open by exocytosis to the lumen. This is an EM picture of the Golgi apparatus. Its nickname is the secretory vesicle. As you know, the Golgi apparatus is formed of stack of saucer-shaped sacules. We have two surfaces, the cis through which the transfer vesicle goes into the Golgi apparatus comes from the rough endoplasmic reticulum, and the trans phase at which the secretory vesicle arises. Okay. So this organelle is EM picture of Golgi apparatus. The nickname is secretory vesicle. The yellow star marks the stack of saucer-shaped vesicles. One visible feature, we have two surfaces. It appears thin at the center and dilated at the periphery. The blue arrow is the cytoplasm. The red arrow is the transfer vesicle that goes to the cis phase, while the green arrow points to secretory vesicle. This, dear students, an EM picture of a centriole, from where you know 9 triplets of microtubules. The red arrow is the microtubule, and these are 9 triplets of microtubule, the visible feature of the centriole. The centriole is an important non-membranous organelle, important for cell division, stained with iron hematoxylin. From the centriole, cilia arise, flagella. This is a EM picture of the shaft of a cilia. What is the visible feature of identification? 9 doublets of microtubules and 2 singlets surrounded with cell membrane. The organelle is the shaft of the cilia. The yellow arrow points to the 9 doublets, while the blue arrow points to the singlet. This is an EM picture showing the red square surrounding a shaft of a cilia, while the blue square surrounding TS section in the microvilli. This comes from a cell in the inner ear you're going to study, inshallah, in second year. The red square surrounds the shaft of the cilia. Give one character formed of 9 doublets surrounding 2 singlets and they're surrounded with the cell membrane. The blue arrow points to the singlet, the red arrow points to the doublets. The blue square points to microvilli, which is formed of actin filaments surrounded with cell membrane, and the yellow arrow points to the cell membrane. This is inclusion. We have either pigments, which is not important, as the carotene, lipofuscin, and tattoo marks. We have important pigments as the melanin, the myoglobin, and the hemoglobin, and we have stored food. These two photos for the stored food. Storage of glucose in the form of glycogen in very active organs as the liver and the muscle. Here is the glycogen granules present in the liver. It's when the glycogen stains by hematoxylin eosine, it will dissolve, so it needs special stain, which is the Beth carmine and the PAS stain. Identify A, glycogen. Identify B, this is a lipid. The lipid here is a unilocular fat lobule in a fat cell. Name the stain for A, best carmine or the pass. Here is the Sudan 3. Visible feature, the glycogen appears as acidophilic or pink granules. Here appears as a orange droplet, a large droplet, a lipid droplet. Figure A present in the liver cell and the B from fat cell or adipocyte. This is an EM picture of heterochromatic nucleus. Dear students, this is the surrounding cell, this is the cell membrane, and this is the nuclear membrane. This is the nucleus occupying most of the cell. This comes from small lymphocyte. Here, the heterochromatin, you should understand something important here, dear students, that the chromatin appears dark with osmic acid and appears basophilic with the hematoxylin. When it is inactive, condensed, the osmic acid stain it darkly black. When it is active, it appears gray. Okay. When it's stained with hematoxylin, when it is inactive, appear condensed, and when it is active, appears as pale with prominent nucleoli. Here is most of the chromatin is the condensed heterochromatin. The red arrow points to the peripheral chromatin, while the yellow arrow is nucleolus associated, while the green arrow the chromatin islands, while the yellow star either a chromatin or the nuclear sap. And this blue arrow is the nuclear membrane. Again, identify the structure EM heterochromatic nucleus. The red arrow, peripheral chromatin, the green arrow, islets of chromatin, the yellow arrow, nucleolus associated, the yellow star, either a chromatin or nuclear sap. The arrowhead is the nuclear membrane. Here to students an EM picture of euchromatic nucleus. From where I know, most of the chromatin appears pale. The black arrow is the nuclear membrane. The blue arrowhead is the nuclear pore. The red arrow is the peripheral chromatin. The yellow arrow is the nucleolus. And the green arrow is the islands. And the yellow star is the euchromatin or the nuclear sap. Identify the structure and be specific EM, euchromatic nucleus. Identify the red arrow, peripheral chromatin, green arrow, chromatin islands, yellow arrow, nucleolus, yellow star, chromatin or nuclear sap. The black arrow is the nuclear membrane and the arrowhead is the nuclear pore. The second chapter in the module 101 is the epithelial tissue. As you know, dear students, that your body is formed of 4 basic tissues, which is the epithelial tissue, connective tissue, muscle tissue, and nervous tissue. The most important issue in understanding the slides of the epithelium is to know whether the lining epithelium is simple or stratified. What do I mean by simple? Single layer. So the first issue, it is a single layer. Then look to the nucleus. If it is flat, it is simple squamous epithelium. So it is one layer of flat cell with flat nuclei, so it is simple squamous epithelium. Okay, the arrowhead points to the nucleus and the yellow arrow points to the basement membrane. The other important issue in the epithelium, I should know where could I find the simple squamous epithelium. Remember 2 words, thin and smooth. Thin and smooth. Smooth for rapid blood flow on the blood vessels and in the serous membranes as the pleura, pericardium, peritoneum. And thin for easy gas exchange as in the lung alveoli and in the kidney in the glomeruli as here. You should not know the where it's present here. I'm not going to ask you, this is simple squamous epithelium present where in this slide. okay? Because most of these slides you're going to study it inshallah in the second year. You should know it is a simple squamous epithelium, and you should know the sites, any sites of the epithelium. So identify the type of epithelium surrounded by the rectangle is simple squamous epithelium. The arrowhead points to the squamous, to the nucleus, which appears flat. The yellow arrow points to the basement membrane. Here is a simple cubical epithelium. From where I know it is a single layer, the nucleus is rounded. Cube means that the length equal to the height and the breadth is the same. Square appears square. Length equal width and the nucleus is rounded and central. Name the structure pointed by the arrowhead, the nucleus. Feature it is central and rounded, and the yellow is the basement membrane. About the simple cubical epithelium, we should study two words, secretion and reabsorption. Secretion, so it lines any, as he lines any glands. Reabsorption means the renal tubules. Here is two photos. A points to simple squamous epithelium, from where if I can see flat cell, simple single layer of flat cell with flat nuclei. The blue arrow points to the nucleus, one feature flat. Identify the epithelium B simple cubical epithelium, the length equal to the width, and the nucleus is central and rounded. Give one feature of the red arrow, central rounded nuclei. Here is one layer. This is the basement membrane, one layer of tall cell, the length more than the width, and the nucleus is oval and basal. This is simple columnar epithelium. Simple columnar epithelium. One feature, the length more than the width, and the nucleus is oval and basal. Okay. Simple columnar epithelium, please remember secretion and absorption as stomach, small intestine. Remember also, if I'm going to absorb, I'm going to have microvilli on the surface to increase the surface area. Another modification of the simple columnar epithelium is goblet cell that produce mucus, and this goblet cell present in the digestive system and in the respiratory system. Another modification is simple columnar ciliated. This cilia present on the surface, and this is important for movement of particles in one direction, as that present in the fallopian tube and the uterus in the female genital system, and the lung bronchioles in the respiratory system. Again, this is simple columnar epithelium. This is pseudo-stratified columnar ciliated with goblet cell. What do I mean by pseudo-stratified? You are a liar. You are not stratified. You are simple. Why I said you looks like stratified? Because there are many layers of nucleus. Why there are many layers of nucleus? Because the cells are crowded, but all of them rest on the same basement membrane. Not all of them reach to the surface. Cilia, because the cells have cilia, hair-like projection on the surface, and contains goblet cell, which appears as a flask shape, the secreting mucus, which appears vacuolated. This present in the respiratory system, in the trachea and bronchus, to sweep out mucus trapped particles. Identify the epithelium, pseudo-stratified columnar ciliated with goblet cell. Two visible features. Here is the goblet cell and here is the cilia. The blue arrow, the cilia, the green arrow, the basement membrane. The cell pointed by the red arrow is the goblet cell, and the star is the underlying connective tissue. As you can see here, the connective tissue contains many blood vessels. Why? Because the epithelium is avascular, takes in nutrition from the underlying connective tissue by diffusion. Here is keratinized stratified squamous epithelium. This is keratinized stratified squamous epithelium. From where I know, I'm having wavy basement membrane. The first cells have basal oval nuclei, then many layers of rounded nuclei, then flat nuclei. Then here is acidophilic ribbon, okay, arranged in bundle, which is the horny layer. So it is keratinized stratified squamous epithelium. The yellow arrow points to the keratin layer. It appears acidophilic or the horny layer. The green arrow points to the intermediate cells, which is polyhedral with central rounded nuclei. The blue star is the connective tissue. Where could I find the keratinized stratified squamous epithelium? It is the most protective epithelium present in your body. It is the outer surface covering the skin and the opening upon which the skin opening as the nostrils or the anus that open on the skin. This is an important slide. You can ask another issue. If we said identify the organ, it is thick skin. If we said identify the epithelium, it is keratinized stratified squamous epithelium. This is non-keratinized stratified squamous epithelium. From where I know, wavy basement membrane, the first layer basal with basal oval nuclei, then polyhedral with central rounded nuclei, then the last layer here is flat cell with flat nuclei. Where could I find this epithelium? It is important for protection at the cornea, vagina, and anal canal. One points to the flat cell of the squamous layer, while the two points to the basal cells. This slide could be mistaken with this slide. This is a stratified epithelium, okay? Its name is the transitional epithelium. Why I could mistake it with the non-keratinized stratified squamous epithelium? Because both of them stratified, okay, and not covered with keratin. What are the points of difference? First, the basement membrane. Here is the basement membrane. Non-clear, non-wavy basement membrane. Second issue, the nucleus here, all of them are rounded. Third issue, the most superficial cells are large, dome-shaped, and sometimes binucleated. The yellow arrow points to the superficial cells, which is dome-shaped, binucleated. The green arrow points to the polyhedral cells or intermediate cell with central rounded nuclei. And the blue star here is the underlying connective tissue. Why we said transitional epithelium? Why the name? Because it lines an organ that has an distended and empty phases, which is the urinary bladder and the ureter. While the bladder is distended, the epithelium become stretched and flattened to have more area for urine to collect. The third chapter is the connective tissue. We said that the connective tissue is formed of cells, fibers, and matrix. According to the matrix, if it is fluid, we are talking about blood. If it is soft, we are talking about connective tissue proper. And if it is firm, we are talking about cartilage. And if it's hard, we're talking about bone. Now you're going to talk about the connective tissue proper. Dear students, as we said before, that the cells are either free or fixed. The fixed cell either UMC, pericyte, fibroblast, fat cell, unilocular and multilocular, macrophage, reticular cell. Okay, the free cell, we have the most cell, the plasma cell, the macrophage, and the blood leukocytes. The fibers, we have the collagen, elastic, and reticular fiber. The connective tissue proper here. This photo is a type of a connective tissue proper, where I can see bundles of acidophilic fibers, which is the collagen fiber. I'm having fibers that move single, which is the elastic fiber. I cannot see the reticular because it should be stained with silver. We have cells, mainly the fibroblast, in the tissue. In the tissue, but not in this slide. The most cell, the macrophage, and the fat cells. It is a loose areolar connective tissue. A loose areolar connective tissue. From where I know, from this star, which is the spaces. These are potential cavities. Name the fiber pointed by the blue arrow. It is the collagen. Why? It is collagen because it appears as a bundle of acidophilic fibers. Name the fiber pointed by the green arrow. It is elastic because it moves single. The arrowhead points to fibroblast. The loose areolar connective tissue is present in all parts of the body except the brain. Why? Because we said this is a potential cavity, can be filled with fluid or gas that can press on the brain in any center, causing severe lesions. That's why the loose areolar connective tissue is not present in the brain. These are two types of free cells. This is the most cell with its basophilic granules and basophilic cytoplasm and active nucleus. The most cells secrete histamine, heparin, eosinophil chemotactic factor, and leukotriene. It has a receptor on the surface for immunoglobulin E. It is stained with metachromatic stain, which is called the tyrosine blue. This is the plasma cell, which arises from the B lymphocyte when become active. The cell is darkly basophilic with negative Golgi image, and the nucleus is cartwheel appearance, and the function is production of antibodies which circulate searching for the antigens. That antigen antibody will join together and the macrophage will eat them, and this is the humoral immunity. These cells around the yellow circle is the most cell, and the green is the plasma cell. The yellow arrow points to the granules, and the green arrow points to the nucleus. The black arrow points to negative Golgi image. Mention two visible characters of the nucleus pointed by the red arrow. It is eccentric, and the nucleus is the chromatin arranged in the form of cartwheel or clock face. This is white adipose connective tissue. White adipose connective tissue. Why? This is unilocular fat cell. This is unilocular fat cell. As you can see here, when stained with hematoxylin and eosine appears as signet ring. And this is the fat present inside the cell. When you want to stain it well, you can stain it with Sudan 3. Two visible characters for the cell: large, oval. The nucleus is flat and the cytoplasm is rim, surrounding an unstained area or dissolved fat. The blue star marks the fat or the dissolved fat, and the red arrow points to the cell membrane. White adipose connective tissue is an important as an heat insulation. As you know that the temperature moves from high to low temperature. The adipose tissue acts as an insulator, keeping your body temperature. It's important to protect support organs. It's important for supporting organs. Okay. This is brown adipose connective tissue formed by multilocular fat cell. It's stained with hematoxin eosine as well as Sudan 3. The cells are small and filled with many lipid droplets. The blue arrow is the nucleus and the black arrow is blood vessel. The brown adipose connective tissue, it's called brown. Why? Because it contains many blood vessels and it contains many mitochondria in the multilocular fat cell, containing many mitochondria containing the cytochrome pigment. The brown adipose connective tissue present in the newborn, then it will be replaced with the white adipose connective tissue. and in adult it will be just present in the interscapular region, mediastinum, and in the axillary region. The function is heat generation. The function is heat generation. This dear student is a type of a connective tissue stained brown with silver. This is reticular connective tissue. The red arrow points to the reticular fibers, which are fine fibers that branch in anastomose. The function of the reticular connective tissue is support, as it forms the stroma of organs. Here, the students is a type of connective tissue in which the elastic fibers are predominating here. It appears single and zigzag and branching. This type is called the yellow elastic connective tissue, appears yellow color in the fresh state, stained brown with or seen. The red arrow demonstrates the elastic fibers. As we said, it appears as single, zigzag, and branch. The elastic connective tissue is important for elasticity. That's why it's present in the blood vessels, arteries, aorta, ligaments, vocal cords, bronchus. This is the regular white fibrous connective tissue. It appears white in fresh state. The black arrow points to the nucleus of the fibroblast or the fibrocyte that form the collagen. The red arrow demonstrates bundles of collagen fibers, which are present in bundles and regular and acidophilic. The regular white fibrous connective tissue is present in places where I can withstand stretch one direction, as that present in the tendons, and also present in the cornea. Why present in the cornea? For protection, as well as to let the light pass in a straight line due to the regular arrangement. This is irregular white fibrous connective tissue, appears white in color. The black arrow points to the nucleus of the fibroblast or the fibrocyte. The blue arrow points to the collagen. It appears irregularly arranged and present in bundles and acidophilic. Where I could find the irregular white fibrous connective tissue in places where I can withstand tension at different directions, for example, the sclera, the perichondrium, the periosteum, the reticular layer of the epidermis. For example, the sclera, I'm having muscles moving the eye upward, downward, to the right, to the left, all this in different directions, so the collagen fibers should be irregularly arranged. The last chapter in 101 is the blood. The blood is a type of a connective tissue in which the matrix is fluid. It's formed of plasma and elements. Why I said elements? Because I'm having red blood corpuscles counting from 4.5 to 5 million per cubic millimeter with variation male from female, and you should study the units. We have platelets, 200 to 200,000 to 400,000 per cubic millimeter, while the white blood cells is from 4,000 to 11,000 per millimeter cubic millimeter. Um, the slides for the blood is called blood film. And all the blood film, you can see RBCs, and all them are stained with leishman. First, the RBCs, it appears with central pallor because the RBCs is biconcave, so the periphery contain more hemoglobin than the center. So the center appears paler. A very important question, how the RBCs adapted to do its function. Biconcave to increase the surface area, not true cell to carry more hemoglobin, containing enzymes for oxygen and carbon dioxide, carrying oxygen and carbon dioxide, plastic to squeeze itself in narrow capillaries, rounded to move in bifurcation, easily in bifurcation. And you should know anemia and polythysemia. Okay, this is a blood film showing this is RBCs with central pallor, and this is herniated RBCs. This is herniated or shrunken RBCs. And the black arrow here points to the platelets. What you can see here is a white blood cell. The white blood cell, we have either granular or aggranular. Depending on what? Depending upon the specific granules. As all the leukocytes, the 5 types of leukocytes contain non-specific granules, which are the lysosome and known as azorophyll granules, while the specific granules present only in the neutrophil, eosinophil, basophil, which are called granular leukocyte. Here I can see granules, so it is granular leukocyte. The granules appears fine and pink, and the nucleus here pale pink, and the nucleus here is segmented, connected together by thin chromatin, so it is neutrophil. Again, the cytoplasm is containing specific granules, which appears fine pink, and here is the segmented nucleus connected by thin chromatin. You should know that the neutrophil count, the percent of the neutrophil is from 60 to 75%. It is the first line of defense. It is a phagocytic cell. Phagocytose any foreign particle, killing the phagocytosed particle by the primary lysosome and the specific granules, which are the phagostin, lactoferrin, and collagenase enzyme. It stimulates the bone marrow to produce more neutrophil, produce monocyte, which is the largest phagocytic cell, and secrete response substance after finish the defense mechanism in order to let healing of the tissue. Increasing the number of the neutrophil known as neutrophilia, and this occurs in acute infection. This, dear students, is a blood film showing RBCs with the central pallor, platelets, and white cell stained by Leishman. This cell is what? Granular or agranular? It is granular as it contains the acidophilic granules. Okay, and the nucleus is bilobed, connected together by thick chromatin. It is eosinophil. It is eosinophil. Normal count is 3 to 5% or 2 to 4%. It releases histaminase, sulfatase, and major basic protein. It terminates the allergic condition either by phagocytosing the antigen antibody and releasing the histaminase that will break the histamine. The major basic protein is important to attack the parasites. Increasing eosinophils more than 5% occurs in allergic condition as bronchial asthma, eczema, parasitic infestation. So this is a blood film stained with Leishman. The blue arrow shows the eosinophil characterized by the acidophilic granules, and the nucleus is bilobed, connected by thick chromatin. The black arrow points to the RBCs with central pallor. The green arrow points to the platelets. Here again, dear students, is a blood film showing is a blood film stained with Leishman. Blood film stained with Leishman. The yellow arrow is RBCs with central pallor. The arrowhead is the platelets. What you can see here, the red arrow. I'm going to see a cell near the size of the RBCs, and the nucleus is dark, and I can see a thin rim of cytoplasm. This is lymphocyte. So this is a blood film stained with Leishman. The red arrow points to lymphocyte characterized by dark nucleus surrounded by thin rim of cytoplasm. The yellow arrow points to RBCs with the central pallor, and the black arrowhead points to the platelets. We have the small lymphocyte. We have T lymphocyte and B lymphocyte. The T lymphocyte, we have either cytotoxic that secrete perforins, the T helper, the T suppressor, and the memory. And the T lymphocyte is responsible for the cell-mediated immunity, while the B lymphocyte when activated by the T helper, change into plasma cell and secrete antibodies, and some of them become memory cell. The B lymphocyte is responsible for humoral immunity. This, dear students, is a blood film stained with Leishman. The blue arrow shows the large cell, which is the monocyte. The nucleus is kidney-shaped. The blue arrow points to the monocyte characterized by the kidney-shaped nucleus, and the cytoplasm appears as a frosted glass appearance. The red arrow points to the monocyte, while the black arrow RBCs with central pallor. The monocyte and the lymphocyte increase in chronic infection. The monocyte is the precursor of all phagocytic cells in the body. When moved outside the blood vessel, it becomes a phagocytic cell in the connective tissue, macrophage, histiocytes, in the lung, dust cell, in the skin, Langerhans cell, in the kidney, in the the liver, the von Kaffer cell. This is a blood film stained with Leishman. The blue arrow shows basophil. From where I know that they are basophil, we have dark basophilic granules obscuring the nucleus. The basophil is the sister of the mast cell. Secrete histamine, heparin, leukotriene, and eosinophil chemotactic factor, both of them initiate allergy. The percent from 0 to 1%. The red arrow points to the neutrophil, which appears granular, fine pink granules. The nucleus is segmented, connected by thin chromatin. And this is the small lymphocyte with the rim of cytoplasm, okay, and darkly stained nucleus. And this is the RBCs with central pallor. This is the bone marrow. This is low magnification. I can see here the fat cells. Here, higher magnification, I can see different blood cells. And this large cell is the megakaryocyte, which is the mother cell for the platelets. Here, dear students, is these are RBCs, and this is reticulocyte. As you can see here, the reticulocyte is a little bit larger than the RBCs. It contains remnants of the rough endoplasmic reticulum or polysomes or ribosomes that appears basophilic. The reticulocyte normally not present in the circulation, not more than 1%. It increases in case of hemorrhage in order the bone marrow release more RBCs to carry the function of the RBCs, but in the immature form. This is the stage that precede the RBCs. This is the full mature RBCs. Here it is considered a little bit less immature because it's not the containing the remnants of the polysomes. Okay, it's stained with a supravital stain called brilliant crescine blue. Brilliant crescine blue. Thank you, dear students.